See if this, there we go. All right, we're ready to rock and roll. All right, so today we're going to talk about glycolysis, which I think is really your first true metabolic pathway in this class that we're going to talk about. I think you did an introduction to metabolism, though, right? You saw a little bit about metabolism. So probably a lot of you have taken biochemistry before, and even maybe even in regular like Bio 101 or something, they might talk a little bit about glycolysis. But I'm probably going to approach it a little differently than what you've seen before. So even if you're familiar with it, it's going to be done a little, we're going to probably do it a little bit differently. So basically, those of you who've had to memorize every enzyme in the pathway before, or all the substrates and all that, I apologize ahead of time because we're not going to do that. So what we're really going to focus on as we go through is the key points. I want you to understand why you do glycolysis, what happens in glycolysis, what you start with, what you end with, what the purpose of the pathway is, and then how it's regulated. Some of the steps, in order to be complete, I'm going to go through the step with you real briefly, but if it's an enzyme that I don't want you to memorize, we're just going to go real brief. There's really 3 key steps in glycolysis, and if you understand those steps, you understand how it works, then you've got all the conceptual knowledge you need and all the knowledge of enzymes you would need for board exams as well. So first of all, what is glycolysis? So it's a pathway, and the textbook that I like anyway, the Lippincotts textbook, has this sort of outline of metabolism here, and you'll see glycolysis and then the TCA cycle are kind of right in the middle. So they're kind of fundamental to all of metabolism. But this pathway of glycolysis, so it's literally glycolysis, we're lysing glucose, is employed by all tissues, and it provides energy, okay, in the form of ATP. We're going to make energy. And actually it makes energy in another way as well, but we'll explain that later. But it directly makes ATP, and then it also forms intermediates for other pathways. So at the end of glycolysis, we're going to end up with pyruvate. It kind of depends on the situation in the cell, but you can convert that pyruvate into something else. And some of the rest of the semester, we'll be talking about what are those other things you can do with pyruvate. We're really going to focus in on glucose today, but other dietary sugars that you're able to metabolize, galactose, for instance, fructose, they will fit into this pathway in one way or another. I can't remember if we have a dedicated lecture on those particular sugars in this class, or you will see them in systems. I don't remember, but you'll see them at some point. So once you understand glycolysis, the other sugars will eventually converge into that pathway. The end product of glycolysis depends on whether or not we have mitochondria and oxygen available. If you don't have mitochondria, or you do have mitochondria but you don't have oxygen, then we're going to convert pyruvate into lactate. But if you are under aerobic circumstances where you have mitochondria and plenty of oxygen, which is the normal situation in most tissues, pyruvate will be the end product of glycolysis, and that pyruvate will be shuttled into something. And we'll spend the rest of biochemistry talking about those some things. One thing to keep in mind as we go through glycolysis, there's going to be some steps that we're actually going to go over real briefly because those steps aren't key regulatory steps, but they're steps where we're going to generate NADH. Okay, that's actually normally a good thing. NADH is the high energy form. So you have NAD plus or NADH. NAD plus is the low energy form. NADH is the high energy form. So normally getting NADH is a good thing because you can derive energy from it. You can actually get ATP from it, as you're going to see by the end of tomorrow. But if you don't have a way of oxidizing that NADH through the electron transport chain that we're going to talk about, then it'll start accumulating and it'll actually inhibit glycolysis. So you actually you require NAD plus in order for glycolysis to proceed. It's an absolute requirement. So you have to oxidize it some way. And that's where the conditions, whether you're aerobic or anaerobic, come into play. If it's aerobic conditions, you'll just oxidize it through the electron transport chain. If it's not, that's where this alternative pathway converting to lactate actually comes in. Okay, so in order to do glycolysis, first thing we got to do is we actually have to get glucose into the cell. So we have a family of glucose transporters, and right now we're talking about sodium-independent facilitated diffusion. So this is just going to go with the concentration. If you've got a high concentration outside the cell and a low concentration in the cell, relatively speaking, you'll get transport. Okay, so this is not an active process. All these GLUT transporters just go with the concentration gradient. So there's about 14 of those transporters, but there's only 4 they consider high yield. If you look at any board review book, these are the 4 that they throw in there. GLUT1 and GLUT3 are kind of the boring ones. They are the basal uptake. They're present in most tissues. You can almost think of them as sort of housekeeping glucose transporters that are always there. Okay, they don't really respond to the environment. They're always there. Most tissues require glucose in some form or another, at least some amount of glucose. And so those ones are going to be relatively stable in terms of the concentration, not really going to vary. And so, especially in tissues where there's a lot of glucose required, they're going to have a lot of those to begin with, and they're going to maintain a lot of those. Things like brain, neural tissue, okay, they're going to have they're going to need a constant supply of glucose. You also, so the 2 tissues that really require glucose, have an absolute requirement, are red blood cells. Okay, that's the only way you can generate energy in red blood cells, and then your brain. Now your brain can't use fatty acids. Okay, it can use ketone bodies, but it can't use 100% ketone bodies. So it always has to have some glucose. And under normal circumstances, it's using almost exclusively glucose. If you get well into starvation, it'll start using ketones. So those 2 tissues require glucose, and as you might imagine, those are very vital tissues, right? So you obviously have to have and maintain glucose available for those tissues at all times. So we also have 2 other transporters, GLUT2 and GLUT4. So GLUT4, this is an insulin-responsive transporter. So when insulin is present, you're going to increase the amount of that on the surface of the cell. So basically, when insulin is present, you're going to transport in more glucose. So hopefully by now you guys realize when insulin's there, it means you're in the well-fed state. That means there's a lot of glucose available. You just ate food and ate some kind of carbohydrates to get the insulin response. So there's extra sugar available. It brings these transporters to the surface, and it kind of carb loads its own cell. So like muscle cells will start accumulating glucose, and they can anything extra they can actually store as glycogen to use later on. The other receptor, the other transporter, GLUT2, this one's actually the real interesting one. Okay, it's present in pancreatic beta cells. That's going to be your glucose sensor. So pancreatic beta cells are where the insulin is going to come from, right? So they're going to do that. But the real interesting thing in terms of trying to think about the model, how things work, is thinking about the liver. So the liver is going to be the one tissue which is going to actually send glucose outside of the cell. Everything else is always just taking glucose in. So muscle cells, neurons, they're very selfish. They just take glucose in. But the liver will take glucose in on certain times and will transport glucose out at certain times. So this GLUT2 transporter actually will transport in both directions. So, and why do you actually have to transport glucose out of the liver? Well, when your blood glucose goes down, that's one of the main jobs of the liver is to maintain blood glucose. So it will actually send glucose outside of the cell. It's really the only cell that does. The kidney does it a little bit as well, but it's easy just for the model to think about how things work, just to focus in on the liver. Okay, does that make sense to you? And if you understand that critical point, you know, that helps with some other things we're going to talk about in metabolism as well. So the liver's got to kind of balance this need to replenish blood glucose, but also deal with large volumes of blood glucose when you eat food. So it's really, and that's partially why they call the liver the nutrient distribution center. It's kind of the brain of nutrition in your body. It kind of decides, you know, how much and what's going to be sent out. Okay, so you also have, in terms of glucose transport, you also have some sodium monosaccharide co-transporter systems. These are actually active transport systems. I'm pretty sure, because somebody asked me a question about this. They thought we covered it in this class, and I don't think we did. Or maybe it was covered in this class, but it was a lecture I wasn't familiar with. But I'm pretty sure you've seen this system before, right? So basically, what you do here is you create, so you want to, in this case, we're trying to concentrate glucose. So we're trying to get more glucose in the cell than is available outside. So you can't use just passive diffusion in that case, right? Because you're going against a gradient. So what you do in this case is you actually form a sodium gradient. So you pump sodium outside of the cell, and that requires energy. So this system requires energy. So you have to use ATP, and you create this sodium gradient, right? And then the sodium wants to come back across the cell, and what you do is you actually transport glucose with it back across. Okay, and so why would you want to do that? Well, there are several tissues. Certainly when you eat sugar, you want to try to get as much of it. Your body tries to get as much of it as it can, so it's going to pump glucose into the cell. And renal tubules in the kidney, you don't want to lose a whole bunch of glucose in the urine, so it's actually going to concentrate it back in. And so there's several places where you would actually want to actually concentrate glucose or go against a gradient. We also have trying to load the cerebrospinal fluid. In cases where your blood sugar gets low, load the cerebrospinal fluid so your brain doesn't run out of sugar. So these transporters are called sodium-dependent glucose transporters. And there's also tissue-specific isoforms of these, but we're not going to go into any specific ones, so we're just going to leave it at that. The GLUTs, I think it's important to know those 4 GLUTs. Those are fair game for board exams, so I would ask you to know those, but we're not going to go into any specific of the SGLTs. Okay, so the reactions of glucose can actually be divided into 2 stages. So we're going to start with glucose, we're going to end with pyruvate. The first 5 reactions are called an energy investment phase. So we're actually going to have to use, ultimately we're going to get ATP from this pathway, but we're actually going to have to use ATP at the beginning. And we use 2 ATP per glucose molecule, and you'll see where that happens. But we end up getting 4 ATP per glucose molecule, so you actually get a net yield of 2 glucose, 2 ATP per glucose molecule as you go through glycolysis. Plus, at the end, you'll have this NADH, which is also a high-energy compound. You can also convert it into more ATP if you're under aerobic circumstances. And then that pyruvate you're left with at the end is a carbon skeleton you can do something with. You can break it down for energy. You can actually convert it into fat. You can convert it into cholesterol. There's multiple different things you can do with that pyruvate. So the end of glycolysis is not the end for what is going to happen to that carbon skeleton. So the actual glycolytic pathway, the first thing we have to do, so we've got glucose inside the cell, right? Now we're going to have to actually phosphorylate the glucose. So This is one of the there's three key steps in glycolysis that you really have to focus in and you really have to know in order to understand the pathway and how it works, and this is one of them, okay, the very first step. And all we're doing is we're phosphorylating glucose. There's two different enzymes, hexokinase and glucokinase. They both perform the same enzymatic reaction, but they're present in different tissues, which I'll explain in 1 second. So what happens when we phosphorylate glucose? Well, in order to transport glucose, it has to be free glucose. Once you phosphorylate it, you've actually made a highly charged molecule. It's very difficult to transport charged molecules, and we don't have a transporter for glucose 6-phosphate or any phosphorylated form of glucose. So once it's phosphorylated, it's trapped inside that cell. Okay? So it's not going to leave that cell. If you wanted to leave that cell, you'd have to dephosphorylate it, which later on you'll see in gluconeogenesis you'll have to do. But so now once you've phosphorylated it, you've trapped it inside the cell. That's the main point of doing this. So I told you we have two different enzymes that are present in different tissues. Hexokinase is going to be the enzyme that's present in most tissues, besides the liver, and actually the pancreatic beta cells as well. But really, to make the model simple, we'll just focus in on the liver. So hexokinase, so all other tissues besides the liver, what do they want to do? They just want glucose for themselves, right? They're going to phosphorylate glucose. They have a high affinity. That means even a small amount of glucose that gets inside the cell is going to immediately phosphorylate it. But they also have a low Vmax. That means they can't deal with huge volumes of glucose. And the peripheral tissues are never going to do that, okay? Your blood glucose is going to be relatively maintained by that liver. It's going to, when your blood glucose goes up from eating a meal, the liver is going to deal with the bulk of that. So the tissues are exposed to elevated glucose, but not like extreme, unless you're in a situation where you can't regulate your blood glucose, like diabetes or something. So they don't need a high Vmax. So they have a very high affinity. Okay, so they can deal with every little bit of glucose that gets in there, boom, it's phosphorylated. But you're not going to have tons of it coming inside the cell. Whereas the glucokinase is going to be the version that's in the liver. Okay, now remember, the liver has to actually take glucose in, and at some point it's got to send it out, right? So how it makes that decision is based on concentrations. But that also means you don't want to have an enzyme with a very high affinity. Okay, because at some point you have to have free glucose available to release out. So if you had a very high affinity enzyme, it would just immediately phosphorylate it and you'd never be able to release glucose. Okay, so it's going to have a low affinity, but it's actually got a very high Vmax. And why is that? Because the liver has to deal with large volumes of glucose when you eat a high carbohydrate meal. Okay, it's going to get the bulk of the glucose is going to be processed through the liver. So it has to deal with huge volumes, but it only deals with glucose when it gets high. Okay? So it's only going to phosphorylate when it gets high. Does that make sense to you? So that's how that first step works. And actually, I went through both of those. I have several slides explaining what I just talked to you about. Okay, so you should know the difference between hexokinase and glucokinase in terms of their enzymatic qualities and why it has those enzymatic qualities in terms of the Km and the Vmax. Okay, so you're going to see as we go through some of these steps, you'll see a slide like this where I don't really have a lot of explanation. I want to be complete. I want you to understand there's more steps than we're really going to focus in on glycolysis. But when you see something like that, I just want you to realize there's an extra step in glycolysis. We understand it's complicated. We don't have to memorize that enzyme, okay? And there's a bunch of enzymes like that. The three that we're going to focus on, the three enzymes that we're going to focus on are all irreversible. So anytime you see an arrow going in both directions, that's not one of the key enzymes, okay? So the glucokinase, hexokinase, that step is our first key step. Here's our second key step in the whole pathway. And this is our rate-limiting committed step. that earlier step, which was actually very important, making that glucose 6-phosphate, at that point, there's actually multiple different things you can do with that. You can actually make glycogen. You could go into the HMP pathway. These are things you're going to see very soon. Once you get past this step, you're committed to glycolysis. So this is the key step for the whole pathway. Those other two key enzymes play a certain amount of role in terms of regulating the pathway, and they're important as well, but this is the big step. This is going to determine, are we doing glycolysis or are we not doing glycolysis? Okay, so phosphofructokinase 1, PFK1, key rate-limiting step for glycolysis. I also will tell you it's probably going to be one of the most complicated enzyme we're going to look at in all of biochemistry in terms of how it's regulated. I don't say that to scare you. I tell you that because if you spend a little bit of time with this and get it straight in your head, which I'm very confident you can do, if you can get this one straight in your head, you can do any one in biochemistry. It's all downhill from here, okay? So I know this one's a little bit complicated and you have to wrap your head around it. You know, if you haven't done it before, you'll have to spend a few minutes with it. But let me walk through how we regulate this enzyme. So if you understand how it's regulated, you understand how you regulate glycolysis. Another important thing before I forget, if I don't say anything otherwise for the rest of glycolysis, I'm really focusing in on the liver. So the liver is going to regulate things a little bit differently than other tissues because the liver has to balance glycolysis with gluconeogenesis, which are exactly going in opposite directions, okay? So it's much more tightly regulated in the liver. So these same types of regulation occur in every tissue, but in the liver, it's much more absolute. And it's really easy, it's much easier for me to think about, and I think for you guys to think about in terms of black and white. I just want you to think glycolysis on, glycolysis off. Don't make it any more complicated than that. Now, I think you guys realize it's going to be a little more subtle than that, but let's just think of it like that, and then you'll get a good grasp of it. This step also requires ATP. The first step, the phosphorylation step we looked at also required ATP, so that was energy investment. Here's the next ATP that's required. Okay, so it requires ATP. This is an irreversible step. The other step we looked at was also irreversible. The other one we focused in on, that glucokinase or hexokinase, irreversible. How do we regulate that? How do we control everything? First, we can regulate by energy status. So you can have allosteric inhibition by ATP. Let's focus on ATP first. Okay, this is weird. This takes a second to wrap your head around, okay? It's very, 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 very weird to have an enzyme which one of the substrates is ATP, but I'm also telling you ATP inhibits the enzyme. I can't think of any other enzyme we're going to see in biochemistry where you see that happen, right? Usually the substrate's going to make the enzyme go faster. So what the heck is going on there? Okay, so glycolysis, its job is to actually generate energy, right? So if you have a lot of energy available, you don't want glycolysis to be active. You want to shut it off. And ATP indicates you have a lot of energy available, okay? Unfortunately, the key step here actually requires ATP. So what happens is the binding site on the active site that's going to bind ATP has a much higher affinity, a lower Km. So you don't need much ATP available, and it'll bind it really tightly. But once the ATP levels start to go up, okay, so if you have a higher level of ATP, you have another binding site, an allosteric binding site somewhere else on the enzyme that when ATP levels get high enough, that binds there, and then it inhibits the enzyme. So that's how you can actually have a substrate literally inhibiting the enzyme, okay? Hopefully that makes sense in your head. One of the things when you think about how this is going to be regulated is think glycolysis equals energy. So low energy, you're going to activate glycolysis. High energy, you're going to shut it off. Okay, that'll help you too. Okay, so high energy, ATP is going to shut it off. AMP is going to be the opposite. AMP indicates low energy. It's actually going to activate it. So it's got an allosteric binding site that's going to activate PFK1 if AMP is high. So those are reciprocal, right? You can see that those are reciprocal. Citrate also regulates the enzyme and actually negatively regulates the enzyme. I know you haven't seen citrate yet, so you'll have to trust me. Citrate's going to be an intermediate in the TCA cycle. And I also want you to think, when you hear TCA cycle, think of energy. We'll see that tomorrow. TCA cycle, energy. Okay? So if you're building up citrate, that means you already have a lot of energy available. Once again, high energy, energy available. You want to shut down glycolysis. You want to shuttle that glucose into something else. Let's make glycogen. Let's make nucleotides. Let's do something else. We need to do something else. Okay, so that's how it's regulated by energy status. So ATP and citrate shut it off. AMP activates it. Okay, so this next part is where things, I think, get a little bit complicated. So we also have this activator molecule that can activate PFK1. So there's a second enzymatic step that's kind of a loop off the side of glycolysis. It's a very minor loop, so you don't shuttle a lot of your glucose through that. It's a very small amount would go in that pathway. So it's not going to affect the stoichiometry of the whole thing significantly. Okay, so it's a branch off the side. And what this actually does is this branch off the side is where you actually produce an activator molecule. So the product of PFK1 is actually fructose 1,6-bisphosphate. This side pathway actually produces fructose 2,6-bisphosphate. The whole purpose of this molecule is an activator molecule for glycolysis. You'll see it's actually an inhibitor molecule also for gluconeogenesis. So it actually reciprocally regulates those two pathways. And why do we even have, why not just directly, so what, this intersects with insulin and glucagon. So insulin is going to activate this enzyme. You're going to produce fructose 2,6-bisphosphate. You're going to activate PFK1. You're going to activate glycolysis. So insulin activates glycolysis because you have a lot of sugar available. You want to send it through glycolysis, right? And then glucagon is going to do exactly the opposite. Okay, so it's going to inhibit this. And you'll notice the model that I've written out, and I think that's as complicated as you need to go. The figure from the textbook shows that it's actually even a little more complicated than that, but I think you don't need to go into that much depth. Just realize insulin's high. We're going to activate this PFK2. We're going to make this activator molecule, fructose 2,6-bisphosphate, and it's going to activate PFK1. So it's an indirect way for insulin to activate the pathway. Or you have exactly the opposite. And so with glucagon. So if glucagon's high, that means you don't have a lot of sugar available. That means the liver doesn't want to be doing glycolysis. It's actually going to have to shift to gluconeogenesis, which is the opposite pathway. And so it's actually going to shut down PFK1. You're not going to make that activator molecule, therefore you're not going to activate PFK1. Okay? Now, I realize this is complicated. You're going to have to spend a little bit of time, and in this whole PowerPoint, this is probably where you're going to have to spend the most time, but it's not that complicated, and I'm sure you can get it. Okay? So just focus in on those two, that activator molecule. And you might, one of the things as you're going through this, you're like, why in the heck is it so complicated? Why did your body develop such a complicated system? Why not just have insulin and glucagon directly activate or deactivate PFK1? That would make more sense, right? Well, unfortunately, we don't make those decisions. The body did, but that activator molecule also reciprocally regulates the gluconeogenic pathway, so that might be one reason why. You can make one molecule that regulates two different pathways reciprocally. So you'll see that when you get to gluconeogenesis. So that's my only explanation why that actually happens. Okay, so the next steps, you'll notice I'm not really talking about this much. We're not going to try to memorize the enzyme. There is another, so this is an aldolase enzyme. what I want you to notice here is we are cleaving a 6-carbon compound into 2 3-carbon compounds. So every step from now on for a single molecule of glucose is doubled. So if you're trying to calculate how we end up with 4 ATP at the end, remember everything we do from now on is doubled. So you're sending each 3-carbon skeleton through the rest of the pathway. Okay? Kind of boring. We're just making an isomer. So that's about how much time I'm going to talk about it. Not that it's not important, but it's not regulated. It doesn't control the pathway. There's no diseases associated with that enzyme. Some of these enzymes are so absolutely critical. If you had a mutation in it, you'd be dead. So there's no human diseases associated with them. This is also not glyceraldehyde 3-phosphate dehydrogenase, not an enzyme you have to memorize, but I want you to kind of just notice what's going on as we're going through here. Here's one place where we're generating NADH, okay? So you need NAD plus for this to go forward. If you don't have NAD plus, this will stop. So remember that, because at the end we got to regenerate that NAD plus. We're also preparing with adding that phosphate group to make ATP in the next step. This step is freely reversible. It's very unusual to see a kinase enzyme where you have a freely reversible step. It's not an enzyme you got to memorize, okay? But we are making ATP here. Remember, this step we're going to make 2 ATP for each glucose because we got 2 3-carbon skeletons we're sending through there. And then the next ATP step, we're going to have 2 3-carbon skeletons, okay? So it's reversible. You'll actually notice that it's actually named backwards from the glycolytic direction, and you see that with a number of the glycolytic enzymes. That's because a long time ago when they figured out these enzymes, in test tubes, they actually drove the reaction in the reverse direction in order to see how it works. So it was discovered in the reverse direction. This is a substrate level phosphorylation. I wanted to mention that too. We're generating ATP directly here. The other way of generating ATP is going to be oxidative phosphorylation, which we'll talk about tomorrow. Once again, kind of boring. We're moving a phosphate group around. It has to happen for glycolysis to proceed, but it's not a key regulatory step. So we're not going to try to memorize that step. Once again, kind of a boring step. It's reversible, enolase. Okay, we're not going to memorize that step. You're welcome to memorize it if you want, but I'm not going to test you on it, okay? Nor will the boards test you on it. Okay, so we had 2 key irreversible steps we've already seen. The hexokinase, glucokinase, PFK1, and all that complicated machinery for how it's regulated. Here's our 3rd key regulatory step. Okay, this is actually where we form pyruvate from PEP. The enzyme that does it is pyruvate kinase. This is also labeled, you'll notice, it's irreversible under physiological circumstances, so it's definitely absolutely labeled backwards. Once again, they discovered the activity because in vitro, in a test tube many years ago, and they made it go backward under physiological circumstances. That can't happen. But it is a key regulatory step. So how is it actually regulated? One is we have feed-forward regulation. So fructose 1,6-bisphosphate, that's actually the product of PFK1, okay? If that starts accumulating, it will actually come down. So all those other steps are freely reversible. They're not regulated. They only move based on how much substrate is accumulating. Okay, substrate and product determines which direction they're moving, okay? So if you're building up fructose 1,6-bisphosphate, the next key regulatory step is this pyruvate kinase. That means that pyruvate kinase is not being active enough so that fructose 1,6-bisphosphate will feed forward, say, hey, we got to get going, we're accumulating all these intermediates, and cause glycolysis to increase. So you have feed-forward regulation by fructose 1,6-bisphosphate. Now you're going to have to keep fructose 1,6-bisphosphate and fructose 2,6-bisphosphate clear in your head. Okay, so don't let those two confuse you. Write them both down when you're studying and make sure you got them both straight because they regulate different enzymes. Okay, so fructose 2,6-bisphosphate regulated PFK1. The product of PFK1 is this 1,6-bisphosphate that actually regulates pyruvate kinase. This is also regulated by covalent modification. So this one is going to be directly regulated by insulin or glucagon. So once again, and without even looking at this, you can think, well, am I going to turn it on with insulin or off with insulin? Glycolysis is going to be active in the liver when glucose is high, right? So when you have a lot of, so when insulin is high. So insulin is actually going to activate this enzyme, and glucagon, conversely, is going to inactivate this enzyme. And it is absolutely critical that sometimes this enzyme is shut off, as you're going to see when you do gluconeogenesis. Okay, so you have that feed-forward regulation, and then you have the regulation by phosphorylation status. Okay, insulin actually dephosphorylates this enzyme, which actually activates it. Okay, so if you focus in on those three enzymes and how they work, how they're regulated, you know all you need to know about glycolysis. And then realize, what you're doing with glycolysis, what you start with, what you end with, what you're generating in there. Okay? That's probably a little different than you've seen glycolysis before, but I promise you that's how you can make sense of it in your head as well. Okay, so in order for glycolysis to proceed, if we want to keep doing glycolysis, we have to regenerate NAD+. There were a couple steps in there. They weren't key steps we spent a lot of time on, but there were steps where you actually needed NAD+ in order to go in the direction of glycolysis. So you have to generate NAD+, regenerate it. Normally this is not an issue because most cells have mitochondria, and most of the time you're under aerobic conditions, and you can simply oxidize that NADH back to NAD+. And that step of oxidizing it, you can actually use to generate more ATP, as we're going to see in the electron transport chain. Okay, so normally that's not an issue. And if that's the circumstance, then you can actually just take that pyruvate, and you would actually consider that the end product of glycolysis. The next step, which we'll see tomorrow, we're going to be going into the TCA cycle. So we'll convert that pyruvate into acetyl-CoA. So that's one possible place where pyruvate can go, to acetyl-CoA. And that's kind of the default normal pathway under unless we have a special condition going on. However, if you have a lack of oxygen, or you don't have mitochondria, like red blood cells don't have mitochondria, right? Or mature red blood cells at least don't have mitochondria. Or if you have a tissue where you have an immediate high demand for energy. You have muscle tissue which is actively exercising, where you need a lot of ATP really quickly. You'll actually convert pyruvate into lactate. Okay, so you have an enzyme called lactate dehydrogenase. You'll notice it goes in both directions. Okay, so we can actually convert lactate into pyruvate or pyruvate into lactate in either direction. It depends on what tissue we're in, which direction we're actually going to go. But what that actually does is it regenerates that NAD+, so glycolysis can proceed. So therefore, in like a red blood cell, for example, you'll continually get glycolysis. You'll convert that pyruvate into lactate to regenerate the NAD+, and you'll actually send the lactate out of the cell. Okay? And then the liver can actually deal with that lactate. So lactate, if you have actively exercising muscle, or you have red blood cells, for whatever reason, when you're getting that lactate produced, unless it's excessive, you can actually send it out into the bloodstream. It'll be taken up by the liver. Once it's taken up by the liver, we can convert it back into pyruvate, and then it can go into all the different pathways we're going to talk about throughout the rest of biochemistry. We can use it to make glucose. We can make acetyl-CoA, use it for energy. We can make fat with it. We can do all kinds of things with it. Okay, once it's back in the liver and back into pyruvate, it's got a whole bunch of different possible fates. Okay. You can also, you can go to acetyl-CoA, you can go to lactate, you can also take this other enzyme, pyruvate carboxylase, which you're going to spend some time on when you talk about gluconeogenesis. and convert pyruvate into oxaloacetate. That's one of the first steps, or the first step in gluconeogenesis, depending on which substrate you're starting with, that you'll be talking about shortly. I think next week. Or, as you're going to see tomorrow, oxaloacetate is actually an intermediate in the TCA cycle. It's something you're going to see over and over and over again, this oxaloacetate. Good, bad, or indifferent, you're going to see it many times. So what's the net energy yield? Under anaerobic conditions, you will actually get 2 ATP per molecule of glucose. You'll get a net of 2 ATP. You invest 2 ATP to start with, you end up with 4. And if you're going, once again, if you're going back through and trying to figure out where those 4 come from, remember each 3-carbon skeleton goes through twice in those ATP generation steps. So you end up with 4. Okay, and since you're converting that pyruvate into lactate and sending it outside of the cell, you don't get anything else. Under aerobic circumstances, though, you'll get those 2 ATP, plus you can take those 2 NADH, oxidize it through the electron transport chain and generate more ATP. And you'll actually can do something with that pyruvate as well in the cell. You can actually convert it into acetyl-CoA, which we'll see tomorrow, and generate more energy through the electron transport chain. We can actually take that acetyl-CoA and build things with it. We can build fats, we can build cholesterol, multiple different things we can do. Okay? Okay, so hormonal regulation of glycolysis, really what we're talking about, it's important to recognize at first glance when we're talking about this, we're talking about gene expression, okay? We're not talking about direct regulation of the enzyme itself on this particular slide. Because you guys know PFK1, we don't directly regulate through insulin. We do indirectly through that PFK2, okay? And we don't directly influence the activity of glucokinase with insulin. We do directly affect pyruvate kinase with insulin or glucagon, right? But right now we're talking about regulation at the gene expression level. And as you might imagine, and we're really talking about the liver. Remember, unless I tell you otherwise, we're talking about the liver. So these key regulatory enzymes, all the other enzymes that are not key enzymes, they have to be around all the time. You're either doing one or the other. You're doing glycolysis or gluconeogenesis, and all the other enzymes that are freely reversible are active in both pathways. So you need high levels of those at all times. But these key enzymes you don't necessarily need high levels of, or you certainly need to adapt to different conditions. And an easy way to think about it when you're trying to understand what I'm talking about is think about, I like to use like a low carb or a high carb diet, think about it. And really, when I say low carb, it's easy to just think, let's just say no carb. Okay, so if you have a no carb diet, okay, so you're never processing any glucose through the liver. You're never really doing glycolysis in the liver. Certainly other tissues are, but not in the liver, right? You would actually downregulate. You would have glucagon would be active all the time, right? Your insulin levels wouldn't go up. Glucagon would be active all the time. And over time, you would actually see a decrease in the basal amounts of these key regulatory enzymes. They would go down over time because you're not doing glycolysis all the time. Whereas conversely, if you have a high carbohydrate diet, for example, for prolonged, and you remember this has to be for in both situations for a prolonged period of time, you would actually increase expression of those enzymes because you're sending a lot of things through that glycolytic pathway. Okay? So once again, we're talking about the three key enzymes, and it's nice because the other thing I like about this slide is it shows you the 3 enzymes you need to know. And it doesn't tell you all the other enzymes, which you don't need to memorize, okay? And it really highlights the key steps, too, in bold. Although just recognize we're talking about gene expression there, not insulin necessarily directly affecting the enzyme itself. As we went through glycolysis, I didn't point it out at the time, but you might have noticed there was a little branch pathway in one of the steps. And that branch pathway actually produces 2,3-BPG, which you guys are intimately familiar with, right? You guys know 2,3-BPG. You've already talked about hemoglobin binding and release. So 2,3-BPG actually stabilizes the T conformation of hemoglobin, which I like to think of squeezing all the oxygen out, right? So it actually increases unloading of oxygen at the peripheral tissues. So it's used in red blood cells to affect hemoglobin. So this pathway, this little branch, once again, just like with PFK1, it's a very minor amount. I think in red blood cells, about 10% might go through there. With PFK2, you're talking about an even smaller percentage, so it doesn't really affect the stoichiometry that much. It's a minor pathway, and it's really only active in red blood cells. That's really the only cell where you're going to use 2,3-BPG. And you guys already know what you use it for. And you'll notice that there's a phosphatase that can inactivate it as well. Okay, so there's a few key intersections with glycolysis in human health that I want to talk about. First, you can have glucokinase mutation. So I know you haven't had a chance to study this yet, so I'll remind you, hexokinase was that enzyme. Hexokinase or glucokinase, they perform the same enzymatic reaction. They're phosphorylating glucose, trapping it inside the inside the cell. Biochemistry's hell, right? It's the cell we're talking about. So anyway, we trapped it inside the cell. Hexokinase is the version that's present in all of your tissues, with the exception of the liver. The liver has this special one, glucokinase. And remember that glucokinase dealt with large volumes of sugar, right? You can have mutations that are going to reduce the activity of glucokinase. So we actually increase the Km, so we decrease the affinity even more than it already is. So it already was a low affinity enzyme, and now you've decreased it even more. Or decrease the Vmax. Remember, this enzyme is supposed to deal with a lot of sugar. So either one of those means it's not going to effectively phosphorylate sugar. So what do you think is going to happen to your blood sugar after a meal? It's going to stay high, right? It's going to stay elevated, and that's going to be a version of diabetes. This is called, and it's actually rare compared to other versions of diabetes, but it's maturity onset diabetes of the young. So you'll see this even in young people. But, so just imagine what's going on there. You eat a high carbohydrate meal, you have all this glucose, the liver is supposed to be phosphorylating it and dealing with it, but it can't, so it remains elevated. You have elevated blood sugar. So after a meal, you have elevated blood sugar for a prolonged period of time, which is diabetes. And it would remain probably, depending on the severity of the mutation, maybe well into fasting. Arsenic poisoning. We're actually going to get to arsenic poisoning again tomorrow when we talk about the TCA cycle. So the primary spot where arsenic poisoning really causes death in humans is enzymes that use lipoic acid, which I'll show you tomorrow. But arsenic also affects a step in glycolysis. So that step where you had, and I blocked it out there, but it was an inorganic phosphate, it can actually substitute for the inorganic phosphate, okay? And bind to that backbone, and it will be spontaneously lost, which is not really that big a deal. The problem is that step was used to generate a phosphate group, which we were going to use to make ATP. So you lose the ability to make ATP in that step. So glycolysis still continues, okay, but you're not getting a net yield of glucose. Okay, so that's what happens to glycolysis during arsenic poisoning. Now, in most cells, besides the red blood cell, you have other ways of generating energy. You can overcome this. But arsenic also works in a different manner, which I'll talk to you about tomorrow. Pyruvate kinase mutation. So that was that last key step. There are known mutations in pyruvate kinase, could affect the stability, could affect the Km. So we could raise the Km, which lowers the affinity, or lower the Vmax. Okay, so it's not working as fast as it should. This particular mutation, these mutation types, really only affect red blood cells. Because they rely so heavily on glycolysis versus other tissues, which can generate energy in other manners, they have an absolute requirement for glycolysis to generate energy. So they're really the only tissue you see being affected by this. So in order to understand what's going to happen in a pyruvate kinase mutation, or anytime you affect the energy production in a red blood cell, you require these pumps to maintain the membrane, and when you start to not get enough energy, you lose the ability to maintain the membrane, and it starts to get misshapen, and what happens is your body recognizes that that's a damaged cell, pulls it out of circulation. So that's an example of a hemolytic anemia. Okay, so that's what pyruvate kinase deficiency causes, a hemolytic anemia. It's the second most common genetic enzyme deficiency that causes a hemolytic anemia. So that's a lot to chew on, right? Now, there's more common hemolytic anemias that are genetic, meaning sickle cell, thalassemias, and there's probably some others as well. But those are hemoglobin mutations, and hemoglobin is not considered an enzyme. Pyruvate kinase certainly is an enzyme. And it can actually look sometimes similar to this glucose 6-phosphate dehydrogenase deficiency, which we will talk about later on this week. So when we get through that, try to compare and contrast those. But the glucose 6-phosphate dehydrogenase deficiency, it'll present a little bit differently, and we'll explain that. But it also produces Heinz bodies, whereas pyruvate kinase doesn't. The only extra thing here about lactate production is that if you get excessive lactate production, if it's kind of in a minor situation, you can get muscle cramping. But if you're talking about like an MI, where you have like the heart using a lot of energy and you don't have oxygen available, it'll start accumulating lactate so fast that it'll build up. You won't be able to get rid of it. And it'll actually start changing the pH of the cell, start causing enzymes to denature, and then you get a necrotic cell. And that's what happens. That's part of the pathological process in an MI. Okay, so that's, I think, all I've got for now. I know that's a lot to digest, so maybe it is good that the TCA cycle and oxidative phosphorylation are tomorrow. So I promise you, though, if you can get that PFK1 enzyme down, you'll be golden. All right? See you guys tomorrow.